Hello and good afternoon friends, welcome to CEC-UGC live lecture. Dear friends, as you know that we are carrying a series on plant development under which we are going to carry out or conduct numerous lectures. When we are talking about the plant development, dear friends, today we have included a very important topic for discussion, and you might be very curious to know what this topic is. In fact, I am very curious to know what is under basically this topic is, as the title of the topic suggests, how life of plant begins. Doesn't it sound like how does a life of a human being begins? Well friends, because plants are the part of the environment, so as the life of the human being, the plants too have life. So today we are going to understand how basically the life of plants begins, and I would like to tell you all that today we have with us in our studios again Dr. Ashish Kumar Nandi. He is professor in School of Life Sciences, JNU. So first of all, I would like to welcome our guest, Dr. Nandi. Hello sir, welcome to the UGC lecture. As I already said that this topic seems to be very interesting, that how life of plants begins. I hope that today also we are going to get very interesting inputs through this lecture.
Okay, thank you very much. I hope you will find interesting what I said today. And as you very rightly said, that a life of a plant sounds like is beginning of a life of a human, and in fact there is not much difference. Both are life, both have basic metabolism, basic mode of reproduction, and the way they exist in nature has a lot similarities. So naturally, since plants are living system, so there has to be life. The life begins, life ends. So today I'll speak how life of a plant begins. Now the main point is where you think the life of a beginning should be considered. Like the same question, like whether you think a chicken came first or the egg came first. So for a discussion purpose, we can assume that egg came first, and we can start discussing how the egg cells, starting from a single cell, how multicellular plant develop and in a seed, and how that life begins. So today again, actually some of you, if you have seen my previous yesterday's lecture, you must have seen how and what are the different ways plants development system differ as well as overlap with the systems in animal. So I will not go through those systems, those discussion in details, but definitely a brief discussion of that, which will also be helpful in today's discussion. So let me begin in today's class. So as I discussed in the previous class, that life of any higher multicellular organism begins from a single cell, and the subject developmental biology tries to understand how the complex organism like a plant or an animal develop starting from a single cell. And a few important features in a plant is entirely different from an animal, and one of the important feature is the plant's life. The development process in a plant's life is a continuous process. That means it is not restricted only in the embryonic stage or the embryo which is shown over here. This is embryo, and then these are seedlings, plant, and the mature plant, and the development continues throughout its life. And even though it's a very continuous life, the plant's overall growth can be divided into small units. Those units are called phytomers, and this one phytomer is one growth at the growing axis and one growth in the lateral axis, and that's called phytomer. So the plant can be composed by combination of phytomers. So it can be a simpler plant or can be a very complex plant with lot of phytomers. The other difference between the plant and animal, which is also quite interesting, is that in case of animal, you can see the stem cells mostly convert into permanent cell, And these permanent cells I am talking about, like muscle, kidney, heart, brain, do not normally develop back into the stem cell completely. However, in plant's life, this switch between the stem cells into permanent cells and switching back to the stem cells is very frequent and very common. So this is what is another difference, and so thus you can always consider a plant is a repetitive growth, and lot of phytomers come together one after another and make a series of phytomers coming together make a whole plant. So these three processes which I told you, like lifelong growth, repetitive growth, and this reversible differentiation, these three processes come together which help a plant to grow sometimes almost indefinitely, and they keep growing big and big and big. So big and bigger. So you can see here the plants which really are very huge and enormous structures. Now coming to the point of discussion today, that really as I have started initiating this discussion, how life of a plant, and I am writing here embryo, begins. So I will start by saying how embryo develops. This figure here, it shows a flower, and this part which is reproductive area of the flower has been enlarged over here, and this structure is called as embryo sac. This embryo sac, in other words, is also called female gametophyte. In female gametophyte, what you see is one cell which is marked as egg cell. This egg cell is eventually give rise to new life, and other cells are accessory cells. Now once the pollen germinates on the stigma, the pollen tubes grow through style, and then it comes close to the embryo sac. So there is an opening over here which is called micropyle, and that pollen tube must grow like this over here, it must grow and fuse. Now what happens that before this fusion, the nucleus in this pollen cell divide and generate two cells. Both of them eventually take part in the process of plant's growth. One of the nucleus fertilizes with egg cell, that gives the embryo, and other cell fertilizes with the central cells, and so three cells combine and fuse together to make something which is later on going to develop into endosperm. So in today we will mostly discuss that what all happens after this first fusion takes place. So these processes, as we go along, we will learn how the cell division continues, what are the factors that controls the cell division, and how eventually a new plant would generate, starting from a single cell. So this is the first cell, and this is something what I have drawn here is the first diploid cell that is generated after fusion of sperm nuclei and egg cells. And this cell is, you can say, also one cell embryo, zygote, and this cell undergo series of division. The very first division is a process which you call as asymmetric division. Asymmetric means this makes two daughter cells, this is upper cell and this is lower cell, or you can say this is apical cell, this is basal cell. And asymmetric means these two cells look very different. The lower cell is much big compared to the upper cell. This basal cell after the first division, what I have drawn here, has a very little role towards the overall growth of a plant. This cell will undergo a few division and will produce a structure which is called as suspensor structure. This suspensor helps holding the growing embryo or seed into the ovary, and these cells also contribute to a certain extent on the root development. Now, if you look into how the progression of the cell division takes place, we will discuss mostly about the upper cell. This upper cell first time undergo a division which is what I call here is a periclinal cell division. Later on I will tell you what I mean by periclinal, but as of now you say that if the cells divide, they will be together next to next and two globes will come together, which will increase the say diameter of the ball, if you say that two balls are coming together. And these cells will continue developing or growing for two more rounds. So, and this growth will be continuous like 2, 4, 8, 16 and so on. At least up to 100 cells or so, usually this cell division, all the cells divide uniformly. Now you see here, you can see here that after the first round of cell division, it made 4 cells, and all the 4 cells all are aligned in a line, and none of them actually made 2 tiers. It is a single tier 4 cells. And eventually then there is the next stage. You can see that each of these 4 cells divided in horizontal axis. So you get now 8 cells, 4 in the upper layer and 4 in the lower layer, and these 8 cells will form like a small globe or like structure. And this figure what I am showing here is based on observation, microscopic observation, then neatly drawn as close as possible to what is seen in nature. So the first one, as I told you, this is the first cell, and then this is a cell where you have 8 cells. So in the 8 cells in this picture, 4 are drawn, 4 are shown to you, and 4 are behind, which you can't see them. So this is a stage when you have 8 cells, they are called octant cell. In the octant cells, all the 8 cells are globular, and you cannot make it out which cell is different or which you cannot distinguish from one cell to other, and they are all similar. The next cell division stage, in the next cell division stage, this 8 cells over here will undergo a division, but this division is asymmetric division again. Asymmetric in the sense, now if you see the cells in the outer side of the globe, cells in the outer side of the globe is drawn here, they are much thinner than the central cells. The upper cells, the thinner cells are going to make epidermis or the outer covering of the entire plant, and the inner cells which are there in the 4 inner cells and 4 outer cells. So 8 outer cells and 8 inner cells. The 8 outer cells are going to make epidermis for the entire plant, and 8 inner cells are going to make the entire plant organ, all the tissues, root, shoot and all the inner tissues. And this is the figure which is actual figure of a journal paper. You can see the cells how they are arranged. This is the outer thinner cells, and this is inner relatively globular cell. So this cell division will continue, and again a 16th cell will give rise 32 cells. So again what will happen now, the outer cells, the interesting part of these stages are that once the outer layer is determined, these outer layer cells will never increase the thickness. In the sense, the outer flat layer will remain as a flat single layer throughout the plant, and that layer will form epidermal layer. And the inner cells, you can see, are relatively elongated and arranged in a line. So the inner cells have an arrangement, and this arrangement you can see, if you see this arrow as am I moving as a axis of development, the inner cells are arranged along in this axis. So this is called axialization. The cells are axialized on the growth axis, except the dermal cells, or specifically you should say the predermal cells or dermatogen cells. And in the next stage, what we will find that these 32 cells will divide again and will give rise 64 cell or so. And this is the time you can see the embryo. If you see, this is the embryo, a growing embryo. In this growing embryo now you have a many different kind of cells. There are some cells which are outside, they are flatter. Some cells which are in the center, they are long, and again some cells in the top, they are more like globular cell. So in this stage, if you draw a line now horizontally, or if you draw a horizontal axis, what you will find that there is a difference in the type of cells what you see in the upper layer versus the cells which are in the down layer. And these kind of differences from one part of the any organism, for this matter now in embryo, one part looks different from the other half, it is called as polarization. So in this stage the cells are arranged and polarized. So the total embryo is polarized, one half look different from the other half. These lower cells which you can see here, they are still elongated, they do not divide, and these are the cells eventually will give rise to vascular tissues. So vascular tissues are the conducting tissue that contains xylem, phloem, and this xylem and phloem will develop from these inner cells, inner longitudinal cells, long cells which are arranged along the axis. Now if you note down here in the cells down, these down cells actually arose from the very first cell division which took place after the zygote formation, and these only the cells came up from the original basal cells. These original basal cells or the suspensor cells have no division in this axis so far. That means all of them grown in only one single axis, no further subdivision in the sides. However, each and every cell is quite big, much bigger than what you see the cells in the embryo proper, and this big cells helps to hold embryo in its position. This cell again, which I am showing the arrow here, this cell is the uppermost cell of the lower cells. This is a cell and only cell from this lower file will have some contribution towards the plant's final outcome. In the sense, those cells, there is a term called hypophysis, and this hypophysis contributes to columella and quiescent center which is there in the root. So besides those cells of columella and quiescent center, the lower basal cells have no role in overall plant's development. Now as we go and see the different stages, what you can find that so far what I told you, this up to 64 cell stage, the growth of the embryo became almost all like globe, it was globular. But soon as the cells grew slowly, you can see the structure is no longer globular, this is changing the shape, and this to a certain extent look like triangular now. The cell on the top, there is a flatness and it is much wider than it is in the lower end. In other words, it indicates that the way it began, where all the cells grew almost uniformly throughout, at the later stage they grow differentially. Some part of the cells have grown more, like in these places in the side, whereas certain places grew much less. So where growth is low, the structure will be flattened because the growth is reduced compared to the other organ. Now if the same thing continues, what we will find that the center place where the growth was reduced compared to the side would grow more and compared to the center. So, there will be a cleft formation. This cleft formation actually eventually lead to formation of cotyledons. So, in the growing embryo, this will be one cotyledon and this half will be another cotyledon, and this stage is known as heart stage, because this embryo looks like more of a heart. And the growth continues, and you will see that at the later stage this will form like a full structure, embryo structure, and this too will eventually give rise to cotyledons, and this is vascular bundle, is going to make the vascular bundle, and this is the part which is going to make root, apical meristem or ram root, and this is the final stage of embryo development, what I have told you, I have shown you earlier also. And so many times what happens, plants tries to keep their seed size as compact as possible, unless the seed are like the wind dispersed seeds. So, for many cases the seeds are highly compact, and you can see to make it a fine globular structure, eventually the cotyledons bend to accommodate in the size of a seed. So, this is overall overall cell division pattern, how cell division takes place in, and how from the single cell one seedling develops. So, this cartoon summarizes the series of steps that I have told you so far. So, to have a recapitulation, the zygote forms after fusion of egg and sperm. The zygote undergo asymmetric division. Lower cell, the white cells contribute to suspension. The upper cells now go division, make embryo proper, and eventually this will germinate. After germination will give a seedling, and this seedling which you normally observe in naturally field and so when after germination. So, this is how the life of a plant begins overall at the in terms of cell division. Now, I am showing you the last two stages of development. Here you can see this is an embryo which is called a heart stage embryo, and this heart stage embryo eventually will develop into a more complex structure of plant, and this color codes the contribution of cells. So, you can see this is the green cells here are going to make almost entire above ground tissue, and the cells which are in yellow are going to yellow, and the colors in light pink and blue, these cells are going to make underground tissues. So, this is how the overall ground pattern is. Now, after this initial establishment of life or seedling, for the getting entire plant, the only two ends of this will grow. One is called as shoot apical meristem or SAM, and lower end is called root apical meristem or RAM. So, hereafter the entire plant will grow by dividing from this SAM end or in the root end. So, we have to discuss how this SAM develops and how the RAM develops in a growing embryo, and that is very important question to ask, right, how suddenly the SAM and RAM develop in a growing tissue. So, here I am showing the position of the cells which contribute to the development of shoot apical meristem or SAM, and these cells in the down which I have highlighted here will eventually contribute to the development of root apical meristem. Now, if I have to to discuss about like how really this much complex plant would generate from this simple miniature embryo what I showed you, we need to understand really how shoot apical meristem generates and how this in this growing embryo, how embryo assigns that this is the shoot end, here shoot should come, and this is the root end where root should grow. So, there are many factors, many transcription factors which play very important role in this process. As I told you also in my previous class at the very end, the transcription factors play very important role which eventually can regulate expression of large number of genes. And many of the transcription factors have been identified by their homeotic mutations or change in the organ structure. Today I will speak about mostly the hormonal regulation, how hormones control this development process. So, the most important plant hormone which really contribute towards development, or I would say a polarity, positioning of the organ in the sense where the one branch of a stem should come, where the branch of a root should come, or where in an embryo shoot should arise, is determined by the plant hormone auxin. Auxin must be very familiar to all of you who are watching this. If you have read at least up to higher secondary or school level biology, auxin is the very small plant hormone and that plays very important role. Now, so what auxin do in deciding the positions, I will tell you. And this slide what I am showing you is the different way how people have studied the amount of auxin present in growing embryo sac here, and again later on I will show in the growing embryo. This embryo sac in a cartoon is shown that this end is the micropylar end, because you have egg cells over here and this is the other end. Now, this redness or the red color is something which which says how much auxin you have it. Now, if you look, you will see that auxin concentration is very high in the end where egg cell is lying in the embryo sac. So, there is an inherent auxin gradient in the embryo sac itself, and that inherent gradient is something which eventually going to influence how the embryo develop and how shoot will develop in the embryo. In the next slide, you can see a blue color. This is a real picture. This is a developing, this is an embryo sac inside a flower and microscopic picture. And in this experiment, what is done here is they have taken a gene. You do not have to really worry about the name of the gene. It's UCA1. This gene is required for auxin biosynthesis. So, to know where auxin is accumulated, what has been done here is that the UCA1 gene promoter has been fused with a GUS reporter. So, in the other word, wherever the auxin is synthesizing, the plant will show GUS expression which can be detected through blue color. And you can see in this experiment that you can see that if this is the embryo and this is the micropylar end, most of the auxin is present in this micropylar end of the embryo sac. This is another way for determining and showing the position of auxin, and this experiment has been done by another reporter gene called GFP or green fluorescence protein. This green fluorescence protein or GFP can, when it is expresses, if you look under UV or blue light, it can give you a color, which is a green color. And this has been used, there is also used a promoter, this is a synthetic promoter called DR5. This synthetic promoter turns on when oxygen is present. So, here you can see again that near micropylar end that oxygen concentration is very high. So, what all supports here that there is in the maternal tissue there is a gradient of oxygen. Oxygen is not uniformly present. One part of the cell has much more oxygen, one part of the embryo sac has much more oxygen than the other part. And this oxygen gradient again you can see is something which plays important role in development. And in this cartoon it tells you, shows you where all in the embryo oxygen accumulates. And all the stages of cell development as I showed you is shown here in this process of through these cartoons. So, this is apical cell, this is basal cell, and there are some tiny arrows. If you can see, you will see that oxygen is moving from the basal cell to the apical cell. In other words, later on you will see that lower basal cells do not have much of oxygen. Most of the oxygen is in the upper cells. Here you see those tiny black dots. So these dots represent the oxygen. Interestingly, you will see that up to the 8th cell stage or even 16th cell stage, oxygen is almost uniformly present, and there is not much of difference from the one part to the other part. However, later on you will see oxygens accumulate in the lower half. Oxygen come down and accumulate in the lower half. You see the later stage now, this is very clear, oxygen is almost lost and oxygen concentrate only in the upper cell, what I have told you, hypophysis. Heart cell, now there are 3 points where oxygen is accumulating. Here is one point, here is one point, and here is one point. In other words, actually in the growing embryo, oxygens get transported to 3 ends. So, from here oxygens are pulled here, from here oxygens are pulled here, and from here oxygens are pulled here and oxygens are pulled down. So, oxygens are pulled in 3 different ways, and naturally this area where I have kept my arrow here, you will see this area where oxygen concentration is very, very low, and this is the place where embryo is, where shoot apical meristem is going to begin, and where oxygen is maximum, this is the end where oxygen is maximum, it is going to make root apical meristem or RAM. Now, that pattern of oxygen distribution, what I told you, you can also visualize through this GFP expression under DR5 promoter. You look here, this is the very early stage, oxygen is uniform, oxygen is almost uniform here until almost 32 cell, and then oxygen starts accumulating in the lower cells, and a stage comes when there is no green color in the embryo proper, or not much of oxygen in the embryo proper, all are transported to one corner of the embryo, and this part where it is maximum is going to make root, and this part is here in the next slide. This is the part where its minimum is going to make shoot end. So, at later stage you can see there are some lines over here, you can see these are the lines which are through which oxygens are flowing, and these lines would eventually develop vascular system, and these two will be cotyledon. So, since oxygen, so far I have told you, oxygen shows a very distinct pattern of distribution in the embryo. It is quite high in the root end, very low where shoot apical meristem develops, and most of it is due to distribution of the redistribution of the oxygen. Oxygen biosynthesis takes place in large number of cells, but oxygen but after the biosynthesis, oxygens are getting transported to accumulate in different part of the body differently. And this oxygen flow is very interesting, holds key process development in many cases, and there are two major oxygen transport pathway. One is called long distance transport, or this is something like a highway transport. Here large amount of oxygen can be transported via phloem. So phloem is a network of conducting tissue which conduits from leaf to root and throughout the stems. So oxygen, large amount of oxygen, or say most of the oxygen can very rapidly get transported through oxygen, and this rate is also quite high. As high as say almost 15 to 20 centimeter it can travel in 1 hour. Okay, you don't have to compare with the cars, but you have, if you compare the hormone transport, 20 centimeter per hour, it's quite high. Beside this high way transport, there are local transport, neighbor to neighbor transport, and that neighbor to neighbor transport takes place through a mechanism which requires transporter again. It is not open transport. This is something cannot be diffused completely or transported back. So I'll few minutes to discuss how oxygen are transported from cell to cell. So this cartoon here I am showing like the two different type of transport. One is a high way transport, or this transport is called also non-polar transport. Non-polar means they cannot direct the flow of transport. This transport is dependent on the phloem flow. As the phloem flows, the oxygen will flow. In the other words, there are ones which flow from cell to cell. These are cell to cell or directional transport, or called polar transport. There is something again quite interesting about oxygen. Normally you will see that many of the morphogens, gradients or even minerals, nutrients, everything tend to transport in the way there where you have less, the molecules go there. But in case of oxygen, it's the other way around. And if you see, like in these cells, the blue color is shown as the oxygen, and this arrow shows oxygen flow. If these cells have more oxygen, the nearby cells will put their oxygen transporter in a way so that more oxygen can be transported to these cells. So it forms a gradient. This oxygen is a small hormone molecule. This is a weak acid called indole acetic acid. This the pH of this or pK value of this acid is 4.75. And quite interestingly, what we will find that this acid or indole acetic acid can remain in the IAA plus form or in the IAAH form. So, and that depends on the pH. pH of the cell is high, around 7 to 7.2 to 7.5 even, but inner higher this this outer place which is called as apoplast have a pH quite low, 5.5 to 5.8. So in the low pH there are more hydrogen ion and it forms IAAH. In the higher pH hydrogen ion is low and it makes the IAA form. So this is a polar form and this is a non-polar form. And if you look at the transport in the lower pH, it is in the high protonated form and this is uncharged. So it can pass it through the plasma membrane quite well, which is also largely non-polar, and which is inside, after it goes to inside, it becomes deprotonated or charged. Once it's charged, it cannot transport back. So transport is passive. This is called passive transport. There is no transporter, and this passive transport is one way. So it can transport from one end and it cannot revert back. So there are large number of proteins. These proteins are called oxygen transporter protein. Okay. So this auxin transporter protein, there are quite many of them. Many of them there are called as PIN. PIN is the name is from a phenotype that I will tell you later on, and this is a transporter that transport the cellular auxin to outside. So you can see there is a flow of auxin from one end, it is coming, and then one end is going out. So the auxin flowing from in a directional way. And so if you have two cells next to next, and then auxin make a continuous chain of progress from one cell to other, and which is called polar transport, which is very slow but very effective in development. So what we have told you that auxin must not accumulate in the embryo where shoot apical meristem has to develop, because auxin actually inhibit the formation of shoot apical meristem. There are large number of genes as transcription factor. I told you some of the transcription factor turns on only when auxin level goes really below certain threshold level. So in that cases auxin cannot initiate shoot apical meristem formation. However, even though auxin is not required, or in other words auxin inhibit the formation of shoot apical meristem, if auxin is not there, then shoot cannot give rise to any leaf. So that is very interesting situation where auxin must be absent for making shoot end, but again auxin should come up with a good concentration so that leaves can be made on shoot. So here if you look, there are two figures I am showing. The left figure is from a plant Arabidopsis, and that is a shoot apical meristem. These tiny structures, and this photograph is taken by a technique called electron microscopy, and through electron microscopy you can see these tiny leaves which are coming up. Now the PIN mutant, PIN gene I told you, PIN gene is important for making auxin gradient. Now you see if PIN is mutated means functionally function is not there, then what happens, it doesn't make those leaves. So you can see this is a bare shoot tip. In the shoot tip has no leaves, and this shoot tip look like a pin head. So the gene name came from the pinhead like phenotype of this mutant. So this is auxin which is must be transported properly to make a shoot. This is also very nicely demonstrated in experimentally. Like here in this experiment, what has been done is, this is a pin mutant as I showed you earlier, which doesn't make any kind of any organ in the side. Now in this experiment you can see this red color, this is like a small glue. This glue has been mixed with the auxin. Now if this glue is stuck on this pin mutant, so this is the pin, in one side here the auxin is applied with the help of the glue. Now if you keep it for a few days, you will see there is a differential growth. Now pin has a kind of a uniform growth. Now you see this growth, there is a differential growth, and which eventually will give rise a lateral organ, or other words leaf. So this experiment tells you that the mutant experiment told you that auxin or local maxima auxin is required for organogenesis, and artificial this experiment or exogenous application of auxin tells you that local auxin maxima is sufficient for organogenesis. So it is required and sufficient to make organ on the shoot. So this is what actually I was telling you, that auxin should not be there where shoot is to come up, but again at the same time auxin should be there when leaf has to come up. So that is quite interesting altogether. Now this is what I showed you by the mutation phenotype and exogenous application. Now let us see really how this pin protein, as I told you, pin are the ones which can transport oxygen, how they move in the cell. So for looking into this visualization, again you can use the GFP protein. So GFP can be fused with pin. So wherever pin is there, it will give you a green color. So this is the construct like this. So you have a pin promoter. Promoter of a gene will tell you where the gene should express. So this pin promoter will ensure the pin would express in the place where original pin should. However, this is not a natural pin only. This pin also is having another protein. This is called tagging. So now GFP has been tagged with PN1. So wherever pin is expressing, this will give you green color, fluorescent color. So, and this is the experiment. See this very lovely green color development on the shoot apical meristem. You see, this is the shoot apical meristem where there are two ends. You can see where these green colors are developing, and this is the area where oxygen is accumulating to generate more and more leaves. This color, this figure is much better in the sense you can see this is the shoot tip, and you can see many different leaves which are about to come one after another, and wherever a leaf is about to come, you can see the pins are more, and these pins are present there, which is also diverting oxygen over there. Now you can also, if you expand the figures, you can still see that this pin is localized in a place where leaf organ is going to come, and this is the most expanded figure you can see clearly. But interestingly, again you look, this is not again arranged randomly. This is arranged in a fashion that all the arrows are centered towards a place which is center of the new leaf. So this is the cartoon which shows over here. If this is the I stands for a place where a new leaf will be coming out. It's called incipient primordium. Then you will see all the neighboring cells will arrange their pin in such a way that this is the place where every cell will put their oxygen in. In other words, you will have a lot of oxygen in this place where a new leaf is going to come out. And this is what is also supported by exogenous application. You can give a lot of oxygen, and that will make a shoot end. And once this is formed, then there will be new place where new leaf will be coming out, and this is what continuously happens. So one leaf forms, oxygen goes there, then another new leaf form in a different place, oxygen again start moving over there. So this is the experiment was, I told you, in case of leaf development in case of most of the dicot plants. Many monocot plants, cereal plants, have a different leaf development pattern. In those plants, you will see there are some cells which are called founder cells. These founder cells in the circumference of the stem, and these cells give rise to a new leaf. And quite interestingly, experiments also showed that in those grass plants also new leaf formations requires oxygen. So this is one experiment again where you can see this is a growing tip of maize or corn plant, and this is the shoot tip, and you have many leaves which are coming after one after another. So this is leaf, one after another leaf are coming up, leaf are numbered 1, 2, 3, 4, 5, 6, 7, 8 and so on. Now the experiment what is shown over here is that if you apply one inhibitor of oxygen, so this oxygen inhibitor will not allow the oxygen to transport. It is something like again genetic modification of PIN1. What we will find that there will be not much of leaf, only it will make up to 4 leaf, then it will stop making the leaves. So oxygen is also required in other plants as well. So oxygen is very important making shoot tip for defining the position of shoot tip where shoot has to come, and also defining the peripheral organ where organs should be developed. Now, this auxin develop also influence root development. If you look at the root end in the cartoon here, you will see the auxin comes through the phloem, and this is the place where phloem ends and auxin goes back. So, auxin concentration is maximum here and to the rest of the places. And again this DR5 gus tells you where the auxin concentration is maximum. This is also quite surprising over here. I told you earlier that where auxin is maximum you get root, but apparently auxin has less important role in root specification. In the other words, if you apply NPA, which is an inhibitor of polar transport, there is no much gross change in the root structure. Only what you see that root becomes bigger, and there is some amount of misspecifications. You can see here some cells which are called as columella cells. They make starch, and so this is the columella cells here drawn here. They are the starch making cells. If you give NPA, the starch also is made in other cells. So when you have a lot of more auxins, you get much more amount of starch formation. Starch formation in other cells other than columella also. So maybe I'll discuss about this starch role and all some other time. I just thought that I should also discuss a little bit about the role of other hormones like ethylene. And ethylene also play important role in case of root development. So what I showed you a few minutes back, that auxin, if you have good amount of auxin, there is not much of change in the root structure. However, if you have ethylene more, you can see the root structure gets altered. So this is brief about the root structure. This is the end of the root. Now you can see there are some cells. These are the white cells which are called quiescent center. They are called quiescent center because these are the cells which do not develop rapidly, and these cells hold all the other cells of meristematic cells in the root, and down to it there are 3 to 4 layers of cells which are called columella. This is a paper which came in a research journal that showed that if you have more ethylene, then the overall root structure gets changed. You look in this figure. This is a plant where normal level of ethylene is there. Now if you have more ethylene, then you can see the root structure is altered, and there are a lot of root hairs developed in these sides. It's not only that, what interestingly will say that development of quiescent center and columella cells. So as I told you, in case of Arabidopsis or many other plant, quiescent center, only there are few cells in the quiescent center, not too many. What experimentally has been shown that if you provide a very high level of ethylene, that you can do by exogenous application, like you can apply ethylene is difficult to apply. So ethylene precursor is called ACC, amino carboxy cyclopropane. That precursor can be readily converted into ethylene. So if you apply ethylene more, you can see it will eventually make more of quiescent center cells. So this again shows you that this is wild type, this is one layer of cells. If you have more ethylene, you have more layer of cells, quiescent center cells. And this is not only case of this mutant, other genes also, when there is a more there is a gene mutation called ETO1. So when there is a more ethylene, and it makes more root hair. So actually I am almost end of my talk, and I would like to summarize today what I have told you so far is saying that the life of a plant begins no different way than any other animal or any other system. It begins with a single cell or a zygote. that undergo a very programmed repetitive division of cells, and that cell division is very precisely maintained. And in case of plant, as I told you yesterday, there is no movement of cell during embryogenesis, so cell do not move, rather they are positionally fixed. And in case of plant, the position determines what the cell would be, and in that process eventually it will make a globe, and it will make a structure which is initially globular embryo, that will make it a heart shape embryo, and once the heart shape is made, in the cleft of the heart where the oxygen concentration is least, the shoot apical meristem will form, and in the down of the heart where oxygen concentration is maximum, the root end will form. The oxygen plays very important role in specification of the leaves, lateral organ, and ethylene plays very important role in cell specification in the developing root end. Okay, thank you very much.
With this note, thank you sir, thank you so very much. You have made us aware of how plants begin their life. I hope that all the students who might have watched this lecture or will be watching through the YouTube would be benefited from this particular lecture. With this note, we take your leave, and we like to tell you that in future lectures also we are going to talk more on plant development. As we said that it is basically a series, so every time we will try to bring some new topics for you with Dr. Ashish Nandi. Thank you sir, thank you so very much for being with us.
Thank you.